Hi everyone. In this presentation, I would like to explain about the SIPRA's capability to conduct extractable and leachable studies, and along with this, what is the importance of to conduct the extractable leachable studies, and as well as what are the techniques involved in this process to conduct the extractable and leachable studies. While coming to the in this presentation, I have segregated into the four parts. The first one is the scope and the approach. So extractable E&L studies are meeting the global regulatory expectations. One is the scope and approach. What is the scope of this activity and how we are going to conduct. And second one is the regulatory alignment. So as per current practices, we conduct with this USP 1663 and UN 1664 and any other relevant regulatory guidelines. Coming to the technical capabilities, we had a GCMS, LCMS, MS, ICPMS, and skilled manpower with respect to this technical capability exhibition. And when coming to the quality and compliance, we had a strong QA oversight and data integrity, audit ready documentation. So when coming to the what exactly the SIPRA, so SIPRA stands for a one-stop solution for all the pharmaceutical testing needs. We had a 30 plus years of experience and team size over 100 people and experienced staff on trained GXP environment like this. So what services we provided in this? So SIPRA, in generally we provided with this. So we analytical research, method validation, formulation research, microbiological testing, bioanalytical, preclinical. So when coming to this E&L studies, we focused on this E&L studies. So we had a leadership team. So P. Sriram COO and MDV Satyanarayana, and coming to me, Venkati Adala. I am the SME for technical and quality related execution activities. So when coming to this point, what exactly the extractable, what is leachable? Extractable in the sense to test the material. So whenever if any packing material is contact with your drug product or a drug substance, if you tested with this, we can call it as an extractables. Wherever the leachable, if you store the sample at the normal storage condition to meet the shelf life, so to test the drug product, we can call it as a leachable. So these are the definitions. The extractables are organic and inorganic chemical entities that can be released from a pharmaceutical packaging oblique delivery system into an extraction solvent under laboratory conditions. That means whenever if your drug product or a drug substances is packed with different packaging materials, those packaging materials might extracted through solvent under laboratory conditions. So we're coming to the leachables. Leachables are also same chemicals that migrate from packaging or delivery materials into a drug product during normal storage, use or stability testing. So for which materials E&L studies shall be conducted? So in generally plastic, elastomers and glass container closure systems, medical devices in contact with the drugs, manufacturing and processing material, single-use systems, secondary packing materials. These are the commonly studied. While coming to that, why we have to perform the E&L study? In generally, why we need to perform the E&L study? A scientifically sound leachable assessment is important to manufacturers as leachables can potentially affect the drug product efficacy, safety and quality. So whenever if you are packing material may potentially impact your drug product or a drug substance. So that's why the leachable and extractable assessment is a mandatory for any manufacturer. So additionally, such leachables assessment could provide an understanding of a source of a leachable and how to evaluate and manage leachables during the drug development and manufacturing process. So this will help us to where the leachables might get coming into the product that can be minimized. First one is to ensure the patient safety, maintain the drug quality and suitability, and to meet regulatory requirements, to qualify the selected suitable packing material, to identify potential extractables, to monitor actual leachables during a real-time storage, and to prevent the drug container interactions to ensure product performance and stability, to support shelf life and compatibility studies to avoid toxicological risk from the packing materials. So what are the techniques are involved in this process? The techniques involved are extraction, scouting, discovery, identification, quantification, safety threshold, analytical threshold. The services we offer, so all the extraction, scouting, discovery, identification, all these services we offer for this. So we're coming to the extraction. What is meant by the extraction? So extraction is a process of treating a material with a solvent to remove soluble substances. Extraction is a complex process influenced by the time, temperature, surface area to volume ratio, extraction medium and the phase equilibration of the material. The buffering capacity will be conducted. In a simple terminology, the extraction means you have to be extract from the packaging components. So the extraction techniques are, one is solvent soaking. The simply packing material can soak into the solvents. Then second one is a reflex, and third one is a oxalate operators, and fourth one the sealed vessel, and fifth one is the instrument based solvent, and sixth one is a sonication. So while doing the extraction, these are some critical parameters is there. The chemical nature of the extracting media. So what is the chemical nature of the extracting media? And the second one is the time duration of the extraction process, how much time you are doing for the extraction, the temperature and pressure at which extraction is performed. These are the critical parameters. The extraction medium, the various guidances and recommendations suggest that if the use of a drug product as an extracting solvent is not feasible, then the drug product vehicle or a placebo could be used as an effective extracting medium. Wherever the case is, uh, the medium which is not possible extracting solvents, in those cases we can use drug product vehicle, or a placebo can also be used. The use of multiple solvents with a different polarities, pH, ionic strength, or extracting powers is recommended for high risk dosage form for packing. So these are the some common packing component and possible extraction media. So common packing component, MDA wall elastomer seal. For this we use normally non-aqueous solvents, dichloromethane and isopropanol hexane. Where coming to the dry powder inhaler mouthpiece, we use water and isopropanol. So whereas a small volume parenterals, while rubber stoppers, we use water pH 5.2 and water pH 9.5 and isopropanol is to water 50-50. Where coming to the large volume parenterals, elastic bag. So in those cases, water pH 5.2 and water pH 9.5 and isopropanol is to water 50-50. Where is come to the scouting. Scouting means this is an analytical technique, provide the information regarding the bulk chemical properties of a organic and or inorganic chemical entities present in an extract, which can be used to guide extractables discovery, identification and quantification. So for example, these are the analytical methods commonly used for scouting. One is gravimetric analysis, which will give significant levels of non-volatile residues. And UV absorbance, organic chemical entities are present which the contain UV chromophores within their molecular structure. Infrared spectrum, chemical classes of organic extractables present for aqueous extracts and total organic carbon. Measure the total amount of organic extractables. RY and pH, property of acidic or basic extractables, total amount of non-volatile organic or inorganic extractables. So these are the extractable characterization process. So for example, if you want to characterize any extractable, so you have to discovery, identification and quantification, and then go for inorganic extractables and organic extractables. Inorganic extractables can be done with the ICP-MS, AAS, whereas the organic extractable we can use LCMS, GCMS, GCMS-HS. These are the some common techniques we use. And coming to the leachables, the assessment is performed on the actual drug product, not stimulations thereof. So where coming to the leachables, we perform the activity on the drug product. The assessment is performed within the actual packaging and delivery system. In the form, it will be commercialized not within a prototype or a system component. So once if you pack your product into the certain packing material, we can directly analyze those drug product. Where is the sample preparations, aqueous dosage forms and solid dosage forms. So whereas aqueous dosage forms, leachables can be recovered using liquid-liquid extraction with water-immiscible organic solvents. Whereas a solid dosage forms, leachables can be recovered by dissolving the drug product with an aqueous solution and applying liquid-liquid extraction and extract concentration. Where coming to the scouting analysis, in generally not applied to the leachables characterization, as a drug formula may interfere with the scouting methods. The analytical techniques applied to the leachable characterization are the same as those applied in the extractable characterization, which are summarized and discussed. So where is the coming to the analytical evaluation threshold, how we are going to be calculate the any extractable or leachable is present. This AET establishment is the level at which the unknown extractable and leachable should be identified and quantified in a particular drug product and therefore be used basis of an actual analytical method development. The analytical evaluation threshold is calculated in a simple terminology because the organic and wherever the organic evolutions, you don't have any certain specified limits. In those cases, you have to be evaluate the analytical evaluation threshold AET calculations. In this, AET equal to SCT multiply with SCT means safety concern threshold, which was published in the ICH guideline. So as per this, if you take the LDPE bottle, so then value will be the 1.5 microgram per day. So if you are applying this 1.5, where is a maximum daily dose? So for example, if you are a drug, you are giving maximum daily dose 2 gram per a day. So then you go for it at 2 gram per a day. So number of doses per day. So number of doses per day. For example, 4 times if you are given 1.5 multiply 4 divided by maximum dose of the 2 gram per day. So then where you apply the uncertainty factor, you will get the AET. That means any extractable or a leachable detected above 0.5 microgram must be identified and evaluated for the toxicological risk. So these are the some of the limit for inorganic. So we're coming to the arsenic. If it is comes under the class 1 category, where it is oral PDE, the 15 microgram per day will be the maximum limit, and parenteral it is a 15, and inhalation which is a 2. So these are the some common limits which are already available in the ICH guidelines. So these are the some other elements like tin, antimony, barium. So comes with the class 3 and 2B. So limit of extractables and leachables. There are permitted daily exposure for the limit elemental impurities according to the ICH guidelines. So already I shown you. So whereas there is no standard limit for the organic impurities. Already I told you for organic limits, the completeness of extractable and leachable assessment can only be judged against the overall goal of the assessment. The assessment might be considered complete if all extractable above a defined threshold were identified to least the confident level, the quantitated and correlated both quantitatively and qualitatively with the drug product leachable data and the known ingredients in the packing system, the packing component or a material of construction. So these are the few key timelines where the extraction, how much time it takes. In generally 1 to 3 days, scouting it will take 1 to 3 days, characterization it will take 4 to 7 days, organic extractable and leachables it takes 4 to 7 days, whereas inorganic extractables and leachables it takes within 1 day. These are the some tentative timelines, whereas the extraction process it takes might be stress conditions if you want to apply it might be 30 to 45 days. So these are the some key equipments which are available within our laboratory. So when coming to this GCMS Agilent 7000D triple quad, and the software is Mass Hunter. And where coming to the LCMS, we had a ABSIX 6000 triple quad mass spectroscopy, and the software is called Analyst 1.6.3 software. So we had a very good scientists which have a proven track record and proven executed scientists are available within our laboratory. For this ICP-MS we had a make Agilent and model 7850, which is the most latest one which is available in the market, and we can able to detect 30 element impurities in a single time by using this equipment. So here we use Mass Hunter 5.3, which is the latest version. Where coming to the AS, Perkin-Elman Model Pinnacle 900T auto sampling unit, the software which we use is Syngistix. So all our equipments and softwares, the scientists we trained on these instruments, all are fully capable. They are fully understand the scientific reasons behind the activities. So while coming to this, why we should select the SIPRA? So there are certain things is available with us: 30 plus years of CRO expertise with a strong legacy of quality and trust. The US FDA and WHO, DCA and NABL guidance we follow in our laboratory, and end-to-end testing capabilities under one roof, including end studies, advanced analytical instruments, and high-end softwares. Highly experienced scientific team with proven industry exposure, proven E&L expertise with 100 plus successful studies we completed till now. So coming to the conclusion, so the extractable and leachable studies will be performed at our laboratory in accordance with the applicable regulatory guidelines and industry best practices to ensure patient safety, product quality and compliance. All studies will be executed using validated methodologies and the state of the art analytical techniques. For any clarification, further information, please contact us. So for this, we had inquiries with respect to the ENDL studies or any other validation related studies. So you can contact with these email IDs and phone numbers which are given here. So thank you so much for all your patient listening. If you had any queries with respect to the any kind of analytical needs, kindly contact with these numbers. Okay, thank you so much. If you had any concerns or if you have any doubts regarding this presentation or SIPRA analytical capabilities related queries, please comment below. Thank you. Thank you so much.